thanks everybody. My name is Margaret Murphy. I'm the principal and owner of Integrated Aquatic Sciences. I'll be your moderator today. I'm going to keep introductions very short for all of our presenters so they have the full time to present. We are going to try to keep a strict schedule since we do have 2 other concurrent sessions running with this. So our first speaker today is Thomas Evans from SUNY ESF.
Good morning. Thank you for joining me. So what I'm going to talk about today is the growth of lampreys in 3 different streams within New York. And I can advance things with this. Is that the...
Yes.
No. So actually in New York we have 6 species of lampreys. But for the purposes of the talk today, I'm going to just deal with 2 of them because they're the most widely distributed and also the most abundant within the state. And that's the American Brook lamprey, which is common, especially the Genesee and the Alleghenian up by the St. Lawrence, and then the sea lamprey, which is distributed in a lot of different watersheds throughout the state. So if you don't know anything about sea lamprey, or you want a refresher on it, sea lamprey, which are the most commonly known lamprey, exhibit a life history that's very common for lampreys with a long larval period, and a very weak laser pointer, so let me switch to this, with a long larval period, followed by this juvenile period, which is where the damage occurs if you're worried about fish stocks. The juvenile period is where they're actually feeding on other fish. But I focus primarily here on the larval period since it tends to be relatively long and dominates the life history of these guys. And in this case, I'm showing you that it's 4 years long, but what we're going to see in the talk is whether that's actually true. This is an estimate anyway, or an average about what we might expect to see for sea lamprey in New York. American Brook lamprey are a little bit different from sea lamprey. They don't feed on other fish, right? So they avoid having that juvenile period, and they turn right into an adult after they're done with their larval period. And so their entire life history is almost completely larval, with very short periods where you actually see them swimming around as the adult. And for the work that I did here, one of the questions I want to ask, of course, is how long does it actually take in the populations that I'm looking at in New York State? So this is a very characteristic view of what Ammocoete location might look like. What you have is a lot of sedimentary deposits. There's often mud and sand in that area. There can be buildup of woody debris as well, which is very common for these guys. You might have some buildup of plant material around the side, depending on how stable these environments are through time. And what I actually did was I went out and staked out sites within these 3 different streams, which I'll show you coming up here. And I did multi-class depletions at these sites through intervals of time. So I went out in 2014 and 2015, once each month to these sites. I went from June to October in 2015, and then from May to October in 2015, or sorry, 2014 and then 2015. And all the animals that were captured, I took their length and weight. And this study is a component of a marking study, so this is a small piece of it. But these animals were also marked and then released into the environment. I'm not really going to deal with that today, but I am going to deal just primarily with the growth. So this is what these sites look like. I have sites distributed in the Genesee River, and those are American Brook lamprey. I have a site in the Delaware River drainage, that's sea lamprey, and then another site of sea lamprey in the Hudson River. I think that's probably sufficient for this slide. These are my sampling dates, and that's the black line. So these are actually when I went out and sampled within the year, and the dotted line is the precipitation, the gauge height for a nearby USGS stream gauge. So let's first start with the American Brook lamprey, and then we're going to transition through the other sites. This is a length frequency distribution of American Brook lamprey, and one of the important things to note here is how long they actually might take to grow. And what you can see is that it appears, this is probably the most distinct grouping here, it appears that this is probably year 1, this is probably year 2, and then it looks like year 3. The reason year 3 looks like somebody took a lawnmower to it is because at year 3 they become adults, and they would go out and spawn at that point. Well, the advantage of having this relatively long-term piece of data and also continuous collection over these months is that we can follow these through time, right? So we can follow these peaks through time. So if this is in fact a year 2 peak, then that should appear immediately underneath this peak as it reaches year 2. So if we go back here, I'm just going to use it when it's very clear in July. What we find out is we don't have a year 2 peak, and that appears that that little tiny tail that's sitting off of what I initially called year 2 peak is actually probably year 2. And so these guys are actually probably a conglomerate of year 3, year 4 animals, in fact. And then everything else is probably in the order of 4 to 5 to 6 years old or more. And the reason that I'm so confident that these are actually year 1 is because I collect year 0 animals within these populations fairly frequently. But not terribly different from what you see here, where you have apparently failed years relatively frequently. You can see that there was basically no recruitment in the prior year. So recruitment for these guys appears to be relatively variable, and it may be dependent on how many animals are already present in the streams. They may be limited by the number of animals that are already out there. So if you're actually going to go out and sample lampreys within New York, I'd recommend looking into August and September when you have young of year coming downstream, and also when you have the highest abundance of large animals still remaining. And if you fit a curve to that, so if I'm Bertalanffy Curve here, what you'll see, it's a little bit, the fit here starts to fall off as you get into these larger animals, but in general it's probably taking on the order of 4 or 5, maybe up to 8 years to reach maturity. This dotted line here represents the smallest adult that I captured at this site, so we're probably approaching that. By the time you reach all the way out here, most animals, if not all animals, are probably adults. And this is the number of animals at that site captured through time. Here again is that precipitate, or the gauge height through time. And what I want you mostly to take away from this is that numbers of animals, here's the numbers of animals, and again here's the gauge height, are very much driven by what look like precipitation events, right? So you can have relatively high numbers of animals, and after a single precipitation event, a lot of animals can wash out, and that seems to be characteristic of these habitats, in that these guys are relatively stable within their habitat, but if the right rain events occur, and they occur at the right time of the year, which looks like spring or fall, then these animals will leave that site. Okay, so let's move on to Aquatic Creek, which is sea lamprey, and that's located in the Delaware River drainage. This is a little bit different. So the length distribution here doesn't show a lot of clear peaks, and it looks more like somebody just smeared their finger through the length frequencies. There may be some peaks located here, and I've outlined maybe how much they grow. You can see that they're growing about the same amount between years, right? This is May 2015, this is October 2014. That is, it's hard to say exactly what year that might be, and I know that because there are actually animals down here. I just don't catch them. They're very, very small and hard to actually get a hold of. But those are probably young of here. We're dealing with animals that appear in the population in August and September, and they're about 11 to 12 millimeters long. But at that point, they're so small, it's hard to actually get an idea about how many are out there. I can tell you that in 2014, there were so many that you could have picked up sand in the stream and you would have had 10 or 20 small animals in your hand. In 2015, that was not the case. You would only get a few animals. Again, these are native populations, so they're probably going through these things sort of normally. So if we look at how long it takes, so this, let's say this is throughout the entire growing season, so that the distribution here would give us an idea, right? So there's our young of year. That little tiny black line represents how much these guys might grow within the growing season, and you draw that out. That actually would mean that these guys are about 24 years old. So that seems relatively long, especially for a larval organism, but that's actually not outside of the range of what we know Ammocoetes can live for. We know they can probably live up to a couple of decades. So these numbers up here probably represent, this doesn't mean by default that these are automatically 24-year-old animals. What it means is that if you're in the population, in these populations, and you remain in those populations for your entire life, you will grow very slowly, most likely, and it will take you extremely long periods of time to get to these larger sizes. This population also, I did, you can see here that the captures of animals through time, and then of course the gauge height of a nearby station nearby. The similarity here where you have number of animals declining through time, but you don't have a single large rain event, so it seems relatively steady. On the other hand, in September and October, when we did have rain events, that seemed to drive the population. So again, it looks like these animals are persistent in these habitats during the spring, or during the summer when there aren't rain events. During the fall, they'll probably get washed out. But if there isn't a fall rain to wash them out, they remain there for the remainder of the winter probably. Okay, so let's look at sea lamprey in Cedar Pond Brook. Cedar Pond Brook is a tributary of the Hudson River. And this distribution, so there are fewer numbers of animals here. There's a couple reasons for that. One is I have fewer numbers of dates of these animals actually collected than the other sites. And the other thing is that this site, a lot of the initial sites that I laid out to sample it were washed away while I was working, so that I didn't get access to those later. That occurred primarily between May and June. But I do have a relatively large number of animals, so we can actually draw some length frequencies. And when you do that, these animals throughout May and July, which are probably year one, don't appear to grow very much. They in fact maybe shrink a little bit. These animals entering the population down here are probably young of year, and they appear to be relatively fast growing relative to the other population. So if these are in fact year one, and that would mean that these might be year two, and maybe these are three, and maybe these are year four, and then out here we have larger than four. And if we actually go in and say, okay, well, let's assume these are year zero, and let's measure the distance between that mean peak and the mean peak for year one. So let's say this is the growth that occurs for one full year. And this dotted line now represents the size they need to attain before they can become a juvenile and go out downstream to feed on fishes. Now if we take that measurement, well, lo and behold, if you double it exactly, it lands right on the middle of that peak. And if you triple it, of course, it lands right in the middle of that peak, and so forth and so on. And so that means to actually get out here, you would probably need about 6 years if you're growing at that speed throughout your lifespan. So why don't we have lots of large animals at that site? And if you remember, there was a number of events that occurred not more than 6 years ago that probably washed all those animals out of that site. So we have a number of storm events that probably washed almost everything out of those sites, and so populations in Cedar Pond Brook probably represent a native population of sea lamprey that's recovering from a large-scale event that washed animals downstream in that way. And this is, again, a similar thing to what I showed you before. You can, at this point down here, part of the reason that captures were hard to collect during the middle of summers at Cedar Pond Brook, there are a lot of plants, and sea lampreys grow very well, apparently, in plants. There's sediment underneath them, and so it was very hard to capture these larval sea lamprey because they would be submerged under the plants, and I just couldn't see them. I know that they were there because I would recapture them in September, but I couldn't actually collect them in August. So ignoring this single point down here, what you can see is we don't really have any rain events while I was out sampling in the middle of the summer, and that appears to the population relatively stable with some additions from young of year as those recruit downstream. And here, where there was a rain event, right, there's a large drop in numbers, but that occurs earlier in the summer, spring period. Okay, so the other component of this that I'm going to talk about today is mortality. And this is actually a dead ammocoete that I found at one of my sites. And mortality is a really important driver, right, for determining why you would remain in this site and what are the risks to doing that. And if you estimate mortality for ammocoetes from other studies, so if you go and look at ammocoetes from other studies, what you find is that the mortality is highly variable, right? We can get down to 4%. These are for caged animals in the wild, so there's nothing feeding on them, supposedly. And then you can get probably up to maybe 50 or 60%, depending on what you're doing with different years. And these have been estimated for different species, but not for a lot of species, and certainly not for a lot of locations. So I used a variety of different mortality estimation tools to actually go about estimating the mortality. I'm not going to talk about each one in detail. If you would like to, feel free to talk to me afterwards. I'm also trying to get this published, so if you are interested in seeing it, let me know, and if it gets published, I will send you the paper. But in any case, what you can see is that when you estimate mortality for Dyke Creek, which is American brook lamprey in the Genesee River, you generally have mortalities that are lower. You have maybe your lowest is about 25% up to, this is a catch curve where we have animals, of course, that's being compounded by the fact that animals are washing down the site. But you have estimates all the way up to maybe 95% or 85% if we ignore that catch curve. Oquaga Creek, which is sea lamprey in the Delaware River, appears much lower, and that's partially because their growth appears so much lower. And then at Cedar Pond Brook, mortality appears much, much higher, and that may in fact be related partially again to having that truncated length frequency curve, which may be a result of storm events that are driving them. So the averages here, again, these are relatively large errors associated with them, but I think the averages are meaningful to a degree. The Dyke Creek, again, the American Brook Lamprey appear to have a mortality of around 50, maybe up to 60% per year. Cedar Pond Brook of sea lamprey appear to be about the same, maybe slightly higher year to year. And Aquaga Creek appears very, very low, but again, that may be driven by other things. Those animals are growing very, very slowly, and it may not be, it may be very hard to estimate mortality in those cases. Like I said, that's fairly reasonable based on what we know, probably within the 50% range. So what we've estimated before for Ammocoetes of about 50% mortality per year, this seems relatively reasonable. And that seems relatively reasonable between locations. And unfortunately, it's always hard to predict what pictures will look like on a projector, but you'll have to bear with me. I've tried to point these out with arrows and other signs. What is actually driving that mortality is not clear. I didn't study that directly, but I have a lot of indication that there are animals being fed on regularly. So up here at the top, if we start up here, there's actually a scar on the right, a wound on the back of this animal that's in the shape of a U, right? So it looks like something grasped it at some point and then let go. The animal was able to escape. This darker image, unfortunately, you can't see very well. There's a lot of tumorous growths that are probably related to some sort of pathogen that's occurring within that animal. This animal that looks like I sliced the tail off, I did not actually slice the tail off. That animal actually had its tail bitten off at some point. And then unfortunately you can't see it down here, but this animal has 2 scars on its body that are from puncture wounds. So at some point it was grabbed by something and it left puncture wounds on the body. All right, and with that I would like to thank the Hudson River Foundation for their help in funding this project. And then there were a number of other people that assisted both in the field and also in helping me find locations and information to go out and collect those animals. And I'll take any questions if there's any time left.
We do have a couple minutes if there's any questions. Don't be shy. All right.
There's quite a bit of variability in your estimate between sites, and what do you think the site level factors might be that generate that difference?
I think sites are similar to one another, but they're different in a number of ways. So American Brook Lamprey have smaller reproductive output, and sea lamprey produce much, much larger numbers of offspring. So there will be more mortality if the same numbers of animals are being produced at those sites. The difference also between Cedar Pond Brook probably representing something of a recovering population as opposed to a stable population is also probably driving some of those differences. And it may also be partially dependent on flow and the environment that's around them. Cedar Pond Brook and Dyke Creek are in very, very forested areas with some ag around them. Dyke Creek and Aquaga Creek are forested and ag. Cedar Pond Brook tends to be very, very urbanized around it, but it does support lamprey. So there are probably enough site differences that would drive some of those things, but I don't know specifically which one would do that.
Thank you.
All right, next up we have Andrew Miano, recent graduate now, just defended his thesis from SUNY ESF.
Thank you everyone for coming. Today I'm going to present some work I did as part of my master's project, which I recently defended. Most people here probably know about the round goby, but for those of you that don't know, the round goby is a small benthic fish that's native to the Pondo-Caspian Sea region. And normally in the Great Lakes and where it's invaded, we normally think of them as being around between 40 and 150 millimeters in total length. But recently in the St. Lawrence, we've been finding them in excess of 250 millimeters, which is an extremely large goby. You can see a picture of them on the left there. So a brief history of the round gobies in the St. Lawrence River. They were first introduced around 1990 in the St. Clair River, and then by the year 2000 or so, they've been introduced into all the Great Lakes. And by 2002, there were about 9.9 billion round gobies in western Lake Erie alone. So a lot of biomass. And the first time we actually found them in the St. Lawrence River was in around 2005. In 2006, we picked them up in our long-term surveys at the Thousand Islands Biological Station, and their numbers have been continuing to increase all the way into 2015. So this is long-term data that's coming out of the Thousand Islands Biological Station. The top graph is our trap netting survey, which is actually designed to catch muskie and pike. So we use very large nets, but they actually capture very large gobies. The bottom graph is our 30-foot steining survey. It uses very, very small mesh, and it generally captures a lot smaller gobies. But as you can see, we've seen this really great increase in goby populations over time. And 2015, we actually got the highest numbers of both large and small gobies. So these things are still increasing in our area and are still very problematic. So why actually study the round goby? The round goby does a lot of things in ecosystems. They're a known disease vector. They also displace a lot of native organisms, such as sculpins, log perch, and darters. They're also a known egg predator, particularly on centrarchids, lake trout, and also a saucid, something I touched on last year in my presentation. But they also have this great potential to alter our native food webs. They consume a variety of different prey, mussels, Lethenteron, zooplankton, small fish, and even fish eggs. But it's unclear how these diets actually differ by location or by different size classes, particularly the very large size class that we're seeing in the St. Lawrence River. So our research objectives for this were, does location matter? So are different bays behaving differently in the St. Lawrence River? Are gobies consuming different items? And also, does size or length affect round goby diets? What are these really, really large gobies shown here in the red? What are they doing to the system? What are they feeding on? How much is that going to hurt our ecosystems? So in order to answer this question, we collected 40 round gobies from 3 bays in the St. Lawrence River. We collected 20 large gobies, so these are gobies greater than 130 millimeters, all the way up to 235, and then 20 small round gobies, 48 to 129. And these small gobies are what normally people think of as a regular sized goby. The large gobies are extremely large. And then we analyzed all these gobies for length, weight, diet, dry weight, and abundance, which is more of a snapshot of what they're feeding on, and then also stable isotope content, which is a little bit more integrated approach to see what they're actually feeding on. For our stomach contents, we sorted them all to the lowest identifiable taxonomic group, dried and weighed each of the items, and then we analyzed them using an MDS ordination, an anasim, pairwise test, and we also did a feeding diagram developed by Evanson et al. Our stable isotopes, we used the same 120 round gobies, took tissue from them, sent them to Cornell Stable Isotope Laboratory, dried, weighed, and run through a mass spec. And these isotopes give us, the two we focused on here are carbon and nitrogen. So nitrogen will actually tell you trophic level. The higher up your animal is feeding on the trophic level, the higher nitrogen value you'll have. And carbon, you are what you eat. So your carbon signature will show what that animal is actually feeding on. So the combination of isotopes and a diet will give a pretty good understanding of what these animals are actually eating. So this is dry weight data for our stomach contents. So these are MDS ordinations. So the way you want to look at these is just, if a fish is eating similar to another fish, each of these points is a fish, they'll be very close to each other. And if there's good separation between groups, that means that your 2 groups or 3 groups are eating differently from one another. So our top graph, you can really see that out of our 3 sites, there doesn't really seem to be any differences between the 3 bays that we looked at. And that was confirmed by our anasim as well. A little bit more interestingly is the differences between size masses. The bottom graph there, you can see that there actually is very good, clear separation between our large gobies and our small gobies across all of our sites. And our anasim confirmed that yes, that this is significantly different. But that doesn't actually tell you what large gobies and small gobies are actually eating. Why are they coming out differently? This is average percent weights for large gobies versus small gobies. And a couple things I want to highlight here is that large gobies are really consuming a significantly more dry-scented mussels in their diet, while our smaller gobies are consuming significantly more eggs and also chironomids. So there does seem to be some differences that we can pull out right away just from looking at this data. But this won't actually tell you too much about what they're actually selectively feeding on, whether or not they're specializing on a certain thing or generally feeding on it, which is why we used this diagram developed by Edmondson et al. And what this does is this actually tries to plot out your individual prey items and see whether or not a fish such as our goby is actually feeding specially on something or generally on something. And the way it works is you have a frequency of abundance on your x-axis and your prey-specific abundance on your y-axis. And if, say, you have a goby that all your gobies in your sample are eating chironomids, but chironomids in any one diet don't make up that much of any one diet, then it's going to fall out in a dominant but not important on the lower right-hand corner. But if you have a goby that consumed one very large fish, but no other gobies consumed a very large item like that, a fish would come out as important, but it's not actually a dominant thing that they're feeding on. So we're going to use this going forward. So our smaller round gobies, a couple things we can see. So most items are appearing under rare or generalist. So we can kind of think of as small gobies actually appear to be generalist consumers. The one exception is the chironomids, which most gobies ate chironomids, but in any one sample, they weren't a significant portion of that diet. This is a lot different than our large gobies. Our large gobies, you can see, still have a lot of things that are in the rare or generalist category, but dry-scented mussels really come out in the specialist dominant area. So not only were a lot of large gobies consuming dry-scented mussels, but it was also making up a large portion of those gobies' diets. So they're specializing on those, which is a big difference than our small gobies. The other one exception is, you can see BSB is Brook stickleback, so we actually found a fish inside of one of our large gobies, but it was only in one fish, and it did make up pretty much the whole diet of that fish. So it came out in the upper left-hand corner, so it's important, but not a really dominant prey item. So now that we've got that out of our diet samples, we want to look at our isotope samples and see if this actually agrees. Are larger round gobies actually specializing on dry-scented mussels? This is nitrogen, so this is, you can think of as trophic level. You can see that there really doesn't seem to be too much variation among our sites, but there is this decreasing trend that's significant with larger round goby size. So normally you think of as a larger fish eating higher on the food chain. It's actually the opposite for round gobies. So they're actually feeding lower on our trophic level, which is probably indicative of the fact that they're consuming dry-scented mussels, which are pretty low. What will tell you a little bit more information about what they're feeding on is our carbon. So this is you are what you eat. You can see that really clear trends here that there doesn't really seem to, again, to be much variation among our sites, but between our size classes, you can see that you have this, for our small gobies, it's very, we have a high degree of variability, while our large gobies, it's very truncated. They're all pretty much eating exactly the same thing. And you can see this significant decreasing trend here with increasing round goby length, which plateaus around net minus 22. And published literature and some of our samples as well suggest that minus 22 is about what dry-synods are. So this is a pretty good indication that they are in fact eating on dry-synod mussels. So with taking all these things together, our diet and stable isotopes have, there's very little variation between our sites. So out of our 3 different bay sites, the goby in one bay is going to consume a very similar thing to a goby in a different bay in the St. Lawrence River. But there does seem to be big differences among our two size classes. So our large gobies are the ones that are really specializing on our dreissynid mussels, while our small gobies are actually generalists, but they're the ones that are actually consuming the eggs, at least that we found. So what does this actually mean? Small gobies may actually affect our native ecosystems a lot more than larger round gobies, as they're the ones that are consuming a bunch of different prey items and consuming the eggs. But our large gobies are still going to affect our ecosystems. They're still consuming a lot of different prey items, but they're especially going after these dreissynid mussels. And their populations may actually be related to the number of the dreissynid mussels in a system. You may not actually get these very, very large individuals without dreissynid mussels being present. And with that, I want to thank a whole bunch of people. This project wouldn't have been possible without them and also our financial support. Thanks, and I'll take any questions.
Do you have time for questions?
So, switching diet and size is slightly reminiscent of switch by silvery. And I'm wondering if you looked at, can you hear me all right?
I can hear you a little bit, yeah.
If you look at game differences between small and large categories to see, is the difference when they got big enough to consume the large dreissenids and then they switched over to that diet, or are they just in some other way different strategies?
Yeah, I do think that is a major driver of the difference between the small and the large gobies. The question was about gate limitation and whether or not, as a goby gets a lot larger, whether or not they're feeding more on dreissynid mussels, because they can. I think it may actually be related to the fact that most of our mussels that we actually have left in the St. Lawrence River are the larger quagga mussels, as opposed to our smaller zebra mussels, which have kind of been outcompeted by the quagga mussels. So I think that, yeah, I think the gate limitation is really key. I think that is one of the big reasons. I think once they reach a size that they can consume the dreissynid mussels, they will pretty much exclusively do that.
Yeah, do you have any thoughts on maybe from 30,000 feet for the overall management implications, management for fisheries, management for dreicynids, for large ecosystems or small?
For dealing with the round goby?
Yeah, do you encourage it where you want to get rid of the dreicynids? Do you discourage it? Do you encourage it for lake trout?
Well, once they're really in the system, there's not too much you can do about it. If you do have a system that ends up getting gobies but doesn't end up getting dreissynid mussels, you may not necessarily have to worry about too many very, very, very large individuals. But I think the management is mostly just preventing their spread, especially in areas that have large number of dreissynid mussels anyway, because that's a very dominant food source for them. So those areas that already have established populations of zebra mussels and quagga mussels, those already have a really good food source for gobies. So that's where you would probably want to stop the spread of them.
Maybe you want to encourage them.
But, or, yeah. Yeah. I mean, so we did find that they are specializing on them, but the large gobies are consuming a lot of other things. They're not just exclusively going after the dreissynid mussels, even though they are specializing on them. So large gobies are still a big problem, but they do appear to be really keen into those dreissynid mussels.
Thanks.
Yeah.
I guess sort of watching all these species introductions from afar, one of the things that really struck me with the introduction of zebra mussels was how much productivity was being pulled out of sort of the pelagic zone of the